BioSymetrics condenses months or years of wet lab experimentation into a few days of computational analysis using artificial intelligence and machine learning models. You can gain insight into your phenotypic screen, saving time and money by shortening the time to trial. Researchers can start with any number of compounds, inputting a formatted 2-column file of chemical structures and associated activity. The resulting output will be the mechanism of action we predict for a phenotype of interest. The Progress tab shows our project currently running. We can see that the file has been used to build machine learning models, which predict compound activity for the phenotype of interest. Our sample dataset was used to rank a master list of many thousands of compounds for their probability of causing the phenotype. Many of the compounds in the master list are annotated in public datasets with attributes such as known targets, mechanism of action, or transcriptional response in the L1000 assay. This information is critical for predicting the likely mechanism. When clustering public assay datasets, we identify patterns of similar response among the compounds. By default, we use the CMAP L1000 transcriptional assay and the cell painting morphological assay. Proprietary assays can be added to the workflow at this stage to give further insight. After clustering is complete, the public annotations, clusters, and the master list of compounds are merged to make predictions about likely targets for the phenotype. The description of annotations of compounds is used to give insights into targets that consistently cluster at the top of the resulting expected compounds list. Enrichment analysis of Go annotations predicts targets and target pathways. Let's take a look at the completed project. First, we see the compounds that the machine learning process predicted as being the most likely to cause the phenotype of interest. As we start to scroll, we're able to review each chemical structure. Here, we've identified target genes that are predicted to be influenced by the small molecule compounds. These targets may be important to the mechanism of action for the phenotype, as they may encode proteins relevant to the pathway of interest. Let's select the first target to review the details of the gene and its mapping across different namespaces. We provide a high-level description and the Go terms associated with this gene. For convenience, we also provide the associated links out to various databases where we can review more detail about the gene. Next, we move to the Annotations tab, where Go terms are provided. This helps to identify pathways and processes linking the significant gene targets most likely to be associated with the phenotype of interest. By selecting a bar of the graph, we can see the name of the description of this term, statistics about its significance, and the specific targets found in these results that are members of this pathway. In short, these results provide specific insight into the phenotype of interest, compressing months or years of wet lab experimentation into a few days of computational analysis. For a given phenotype of interest, we can provide a prioritized list of untested compounds likely to cause the phenotype, and supported evidence for the mechanism of action. This allows better lead compounds to be taken forward, and confirmational assays to be planned with more confidence in the expected results. With insights from BioSymetrics, you can reduce the time and expense of bringing a new drug to market.